Welcome everyone. We are going to the last section or the second last section of our Introduction to Food Chemistry lectures. And we want to now understand about enzymes. Enzymes are one of the major proteins that are important in the body. They act as catalysts that speed up chemical reactions and metabolic processes in the body. Enzymes are special proteins that catalyze biochemical reactions, and the substrates bind to the active site of the enzyme, and the enzyme converts the substrate to products without being used. The enzymes can be reused unless inactivated or inhibited. For example, poisoned active sites. So these enzymes, they can always be used to carry out various conversions, and these conversions are done without the enzymes being used up. They remain there. And these enzymes can be activated or inactivated at will. There are certain chemicals that are used to activate, and the others that are used to inhibit or inactivate the enzymes. The enzymes in foods. Enzymes are proteins with catalytic functions. They can be found naturally in food, they can be found in the body, and they can also be deactivated during processing to prevent spoilage. How do enzymes work? The substrate fits into the enzyme's active site. We have the low and key model that suggests that for a substrate to fit into an enzyme's active site, they should be antagonistic. The entry site should be shaped in a way that it is equal to the substrate, so that the substrate can enter into the active site like a lock and key. The way you see the activity of a padlock and the key, that is how the lock and key model operates. And the active site, once has been entered into the substrate, it selectively binds and transforms the substrate, and the enzyme is regenerated after the reaction is complete. What are the factors that may affect enzyme activity? One of the key factors is temperature. Enzymes operate at an optimum temperature range, that is about 30 to 40 degrees Celsius. A higher temperature leads to denaturation, because we know that enzymes are protein in nature, and higher temperatures lead to protein denaturation. pH also can determine the activity of enzymes, and then electrolyte concentration, and finally the shear and pressure. Enzyme denaturation and its applications. Denaturing agents. Some of the agents that lead to protein denaturation, we have heat, pH extremes, and also organic solvents. Example, the polyphenol oxidase leads to enzymatic browning in apples. The applications, amylases can be used in baking and also brewing industries. Here are some food enzymes and their functions in the body or in food processes. Proteases hydrolyze peptide bonds in proteins. Amylases convert starch to sugars. Pectinase breaks down pectin in fruits. And then lipases hydrolyze fats. Lipoxygenase oxidizes polyunsaturated fatty acids. And then cellulases break down cellulose. And then lactase converts lactose to glucose and galactose. Each of these functions will be used specifically according to the kind of reaction you desire to have, or the kind of food product you desire to create. Industrial enzymes, these are enzymes that are added to food. One of them we have rennet. It is obtained from calf, the stomach of a calf, and it's used in cheese making. Microbial enzymes also, such as amylases, proteases, and cellulases are also important, especially during some processes such as fermentation. Proteases, like we have said already, they break the peptide bonds and generate amino acids. Example is pepsin that works in the acidic pH of the stomach. Pancreatic proteases such as trypsin, chymotrypsin, and elastase work in the alkaline intestine. And then there can also be used industrially, such as rennet, papain, and bacterial proteases. Rennet, we said, is extracted from cow's stomach and is used in cheese making. It hydrolyzes the K-casein, that is from PHE 105 to MET 106, and it destabilizes meat cells. It requires calcium ions for the reaction to take place and also an optimal temperature, which converts milk into curds and whey and later forms cheese. The other key enzymes, we already talked about them. The amylase hydrolyzes starch to maltose and glucose. It can be found in the saliva and can also be used industrially for various processes. Pectinase breaks down pectin in fruits and it is used in juice or wine making. Lipase breaks down the fats into fatty acids and glycerol. It can be used in cheese flavoring and also in butter oil. Cellulase breaks down cellulose to glucose and can be used in juices and wines. And lactase breaks down lactose to glucose and galactose. And it is mainly applied in lactose-free products. And these products are very important for people that usually have what we call lactose intolerance, whereby the body does not produce the enzyme that breaks down lactose. So they have to consume lactose-free food products. There are also some enzyme-related quality issues, especially polyphenol oxidase, which causes browning in fruits and vegetables. We do not desire this browning sometimes, especially in the fresh fruits and vegetables. And so we have to devise some methods, inhibition methods. How do we stop this polyphenol oxygenase from causing browning? By heating, by also adding acids such as lemon juice, sulfites, and also removing oxygen. And these are important aspects, especially if you want to preserve your fruits to remain fresh and not to turn color. You have to apply any of these methods according to what you desire from these fruits and vegetables. Lipase in milk can cause off flavors if not inactivated. Lipoxygenase can also cause off flavors in legumes. We also have what we call immobilized enzymes. These are enzymes that are bound to their matrix. For example, alginate beads. These can be reusable and they can also be used to maintain food quality. Example here, we have the immobilized lactase for lactose-free milk. Casein proteins in milk. We already talked about casein when we were talking about milk proteins. And this is the main milk protein. It has the alpha casein, the beta casein, and the K casein, and it forms micelles. The micelles can be stabilized by colloidal calcium phosphate. The K casein forms hydrophilic surface and that provides stability to the milk. The key points to note: enzymes are very essential in food processing and digestion, and controlled use of enzymes leads to improved product quality. Improper use or control leads to spoilage or undesirable traits. And then immobilization and proper conditions can optimize the benefits of enzymes. There are some processes that require you to deactivate the enzymes in order to prevent further processes from occurring after the product has been formed. And this can help to prolong the quality and the shelf life of the product. And there's some enzymes that may be required to produce a better product. So they have to be activated. So it depends on what exactly you require. As long as you have controlled the process well, you will improve the quality of the product and also increase the shelf life. And you'll be able to make a lot of products in the food processing industry. Thank you.